I'm Chris White. I'm the Chief Business Officer at Ocular Therapeutix. I wanted to thank OIS for this opportunity. I'm really grateful to have the chance to speak with you all today. This is a reminder that Ocular Therapeutix is a publicly traded company, and I'll be making some forward-looking statements. So our company develops innovative medicines for a range of ophthalmic diseases by finding better, more predictable ways of delivering drugs to the ocular surface, to the trabecular meshwork, and to the retina. We do this by combining our proprietary polymer technology with established APIs to create durable treatments that provide continuous control over chronic ophthalmic diseases. As we like to say at Ocular, our goal is to make eye drops and immediate release injections a thing of the past. Our product portfolio, which is shown here, illustrates our company's focus on a range of diseases, including wet AMD, diabetic retinopathy, glaucoma, and dry eye disease. We currently market our product Dextenza, which is an intracanalicular formulation of dexamethasone, and it's approved for the management of postoperative pain and inflammation. I'll also note that we're developing an intracameral formulation of travoprost for the management of IOP in patients with glaucoma, and we look forward to sharing phase 2 data on this program early next year. But today, I'll be talking about Axpaxli, which we are now entering our first pivotal trial for the treatment of wet AMD. So first, the details on Axpaxli. We formulate Axitinib in our polymer framework, which is fully bioresorbable. We're leveraging a validated hydrogel platform, which has been used in about 5 million patients to date. Axitinib, as you all know, is a potent tyrosine kinase inhibitor and inhibits all of the isoforms of VEGF. Axpaxli is delivered through an intravitreal injection using a 25-gauge needle and provides continuous drug delivery for 9 to 12 months. This chart illustrates Axpaxli's second phase 1/2 trial in wet AMD patients, and it showcases the product's efficacy. The study involved 20 subjects with active disease. You can see each subject's treatment history, with the bubbles representing injection type and frequency. At week 0, the 15 subjects on top were given an intravitreal injection of Axpaxli. At week 4, all subjects were given a dose of aflibercept 2 milligrams, and the 5 subjects in the control arm were then treated with aflibercept every 8 weeks. The boxes represent patient rescues. So the purple ones were rescues that were not made according to protocol, while the green boxes show the rescues that were according to protocol. So in terms of results, note that the first per protocol subject rescue didn't occur until week 32, and the Axpaxli subjects benefited from a reduction in injection burden of 89%. Taking a closer look at the product's efficacy, the top chart here compares BCVA for the subjects on Axpaxli in blue and aflibercept in orange. As you can see, and as we hoped for, the lines are essentially superimposable. The bottom chart compares central subfield thickness between the two groups. Here again, the lines are very similar. For the aflibercept line in orange, we can see a bit of the sawtooth pattern we would expect to see in a product dosed episodically every 8 weeks. So, how do we plan to get Axpaxli approved by the FDA? We initially considered a standard non-inferiority trial design like the one shown here. However, during subsequent discussions with the FDA, the agency recommended against the use of sham injections. So, for a non-inferiority trial design, we would have needed to introduce a third arm dosed identically and treated the same as the investigational arm. In this design, we would have shown both non-inferiority to the comparator arm, for example, aflibercept 2 milligrams every 8 weeks, as well as superiority to the third control arm. In recent months, we've had lots of discussions with key opinion leaders and regulators to determine what's the best pathway for FDA approval of AXPAXLI. And we've now settled on the superiority trial design shown here. Subjects will be run in on 2 induction doses of aflibercept at week -8 and week -4. Subjects that achieve 20/20 vision at week 0 would then be randomized to receive either a dose of AXPAXLI or aflibercept 2 milligrams. The primary endpoint is the proportion of subjects that maintain visual acuity at 9 months. Rescued subjects will be treated as needed by the investigator. This trial, which we call the SOL trial, is now underway. We have initiated several U.S. trial sites, and our Special Protocol Assessment, or SPA, was approved by the FDA just last month. And we expect to screen our first patient this month. Next quarter, actually early next quarter, we plan to share interim data from the Helios trial of AXPAXLI in patients with diabetic retinopathy. And we plan to initiate the second of the 2 pivotal trials in wet AMD in the second half of next year, second half of 2024. And so, to wrap up, this graphic illustrates the challenges that we see in the management of wet AMD. While patients often see gains in visual acuity during clinical trials, those improvements are typically lost as patients are unable to comply with the rigorous dosing frequency required of anti-VEGF agents. With AXPAXLI, our goal is to provide continuous control and better management of this chronic disease. Thank you all for your time.